What's a really good example of understanding what cells are noisy or the activity of a cell is noisy? Like, what advantage does that give the cell? Is there a good example that you have for like why would the activity of a cell be noisy?
Um, okay, so yeah, that means that be, any molecular system, I mean, I suppose to a chemist, it sort of makes sense that this is the case that we chemists kind of think of, and I trained first as a chemist. We know that chemistry is noisy, that it's stochastic, that things are kind of moving about, with Brownian motion. Whereas, it seemed to me that the biological picture, the old picture, was much more deterministic than that. It actually seemed to deny that stochastic, noisy character of chemical systems, and seemed to be implying that everything was like those animations where everything is kind of moving into the right place. So there is this noise that is intrinsic to the molecular world, and biological systems have to be able to cope with that. And we've talked about some ways in which they're robust against that, but I think it totally stands to reason that actually sometimes biology has found ways of using noisiness. um, and I suppose one example of that is just the way biology can uh, noise is a way of trying things out. So in and not getting stuck in the wrong states. So
Yes.
it's a noise is made use of for classic sort of computational algorithms for optimizing things. There's there's one called Monte Carlo algorithm, which is a way of finding the sort of optimal outcome by sort of trying things out bit by bit. And the way that's kind of done. I'm really describing the process called simulated annealing, where you kind of slowly sort of cool the system down and hope that it's going to converge into its most stable ground state. But you don't want to get stuck in other states along the way that aren't the lowest energy one. And so you make sure there's a bit of noise in there so that it can continue to explore locally and not get stuck in little, local minima, but can gradually find its way down. It's the same process as why we shape the salt cellar, to add a bit of noise so that the salt comes out and doesn't get stuck in some jammed state. So biology can, make use of that too. And I think we see that in developmental systems where, the challenge for a growing embryo growing into a complex organism like us is for all the cells to develop into the right kind of cell type, whether it's a heart muscle cell or neural cells or skin cells or whatever, in the right place at the right time. And that's going to be very noisy. And we can actually see now, because we can track individual cells during development, and we can sort of see them almost exploring the landscape sometimes. And sometimes, they're going as though towards a neural cell fate, for example. And then because of the signals they're getting from cells around them, they kind of realize, to put it that way, that's not the right thing to be at that time in that place. And so they change their mind and they sort of go somewhere else. And you need that noise to have that sort of adaptability, that
Yes.
constant, adaptability. Whereas if it was just following a program, an inevitable program to become a nerve cell, then things are going to get screwed up, and things are going to be out of place all the time.
Yeah.
So yeah, that noisiness is a way of, finding your way to the right place in the landscape and not getting stuck in places you shouldn't be.
Yeah. Like, I think we underestimate the fact that every cell in our body, despite a few examples like sex cells or red blood cells, have the same packaging of DNA, but still our cells in the DNA in our kidney expressed kidney cells, neural cells, pancreas cells, liver cells. Like, that's a phenomenally just we just don't, I think, appreciate that fully. And you're right that I think without the noise, you would just end up with like a Lego character, but different arms, different legs, different, you know what I mean? So all the different pieces would just be jumbled up and it wouldn't be able to know. You have a really good example, I think, in the book where you talk about hematopoietic stem cells in mice, ones that decide to become either red blood cells or white blood cells. The fate decision is largely controlled by two transcription factors, GATA1, PU1, with another player, GATA2, in the mix. And in the biology textbook picture, what you'd imagine is like this tiny genetic toggle. Flip one way, become red blood cell. Flip the other, become white blood cell. But what Steidl and colleagues actually found is that inside real cells, the genes for GATA1, GATA2, and PU1 fire in these sporadic random bursts of transcription. And they found that using fluorescent tags on the RNAs. And they literally saw the cells blinking with noisy pulses of all three, which is just phenomenally beautiful start, even though these factors are meant to be antagonistic to the system. So it looks like chaos, but then when they modeled the system, they realized that the noise actually helped, that those transcriptional fluctuations made it easier for the cells to reliably reach either of the target cell lineages instead of getting stuck in this halfway spot. Noise keeps the fate options open, lets the system explore the landscape, and then tips it into a stable identity. When I read that, I was like, that's so cool.
It
Amazing.
It really is, and thank you, Evan, for getting that very specific example of the kind of general thing I was talking about there. I mean, that's an indication of how, in this book, I've gone into the weeds sometimes.
Yes.
I felt like I've really had to show this is real stuff. This isn't just some metaphorical idea about what cells might do. We see this happening in very particular cases, and there's one. And that's a reflection of, so what you have in a situation like that is a sort of delicate balance, really, that you have. There are what we now talk about and think of as attractors in that landscape of possible states the cell could have, a massive landscape. The number of different genes, combinations of genes that could be on and off in any cell. I mean, it's literally astronomical. And yet somehow, they find their way into a finite set of cell types. And we don't know how many, because we don't quite know how to define a cell type. But, we can meaningfully think about a skin cell being different from a, nerve cell or something. And they find their way into that because that huge dimensional space is somehow kind of condensed by the the biological process into a much lower dimensional landscape with attractors with different cell types in. And the question is how the cells navigate between these attractor states, and that can be described. I talk about it in the book. It can be described using the language of dynamical systems generally, because we're familiar with a sort of complex double pendulum or something. We'll have different attractor states, or the solar system has different attractor states of where the planets orbit. So we're familiar with it from simpler systems, but it seems the same ideas apply in biology, and it makes perfect sense that there would be this landscape of possible states with quite sort of broad basins. You don't have to have exactly these genes on and these genes off, but you have to be in that right sort of vicinity to be that kind of cell type.
Yes.
And there's this balance between that's a kind of deterministic picture where you're drawn into particular attractor types, but also the stochastic picture where you can be exploring the landscape to find them. And what we've now known for sort of 20 years that actually you can tweak cells in what seem to be quite small ways to make them jump from one attractor to another. And in particular, you can make them jump from the attractor of, say, skin cells all the way back to the big, broad attractor of stem cell states, where cells that can develop into any tissue type. And you can do that remarkably easily just by treating cells with 4 or 5 different factors, basically proteins or gene products, to get them to make that sort of journey in the landscape back to a different type. And so this way of thinking helps us to understand how it is that we can reprogram cells to get them to develop into particular tissue types, maybe for regenerative medicine, maybe even for treating cancers.
Yeah. I love these metaphors. I think it's one of the best metaphors that biology has, is this dynamical landscapes of valleys and forks. It's just such a beautiful way, I think, of really conceptualizing what the molecules are doing, what the cells are doing, what the systems are even doing. It's all sort of these dynamical landscapes. Can you kind of explain a little bit more on that? What is this idea of canalization?
So this picture, this landscape picture, goes back to the developmental biologist who called himself an embryologist back then, Conrad Waddington, in the 1940s. And he famously drew this picture of it's like a sort of system. It's like a river drainage network, a system of sort of valleys, that where, the cell types, but it's sort of like a drainage network in reverse, where you start off with one valley and it splits into two and then splits into four as the further down you go, to represent this notion that you start off with a single cell state, stem cells, and then they split into maybe two or three different cell lineages, each of which can become all sorts of different types of tissue. And, they progressively specialize until they reach the mature tissue types, as cell types. So that was Waddington's idea of a developmental landscape. And it was a very influential idea, but it was kind of seen as metaphorical until we got to this modern view of being able to actually see which genes are on and which genes are off in individual cells. And we can actually literally now start to plot out what these landscapes look like, that are, basically the real versions of Waddington's sort of metaphorical ones. So we can actually see what this landscape looks like. And it's more complex than Waddington's. For one thing, you can find some valleys kind of have loops that, come round to the same sort of end state. They're not just continually branching, but, we can actually draw physical pictures of what this looks like. And canalization was a kind of part of that, in that, as the sort of word implies, really, these valleys, they're not narrow, they're quite broad. So you can start from various positions and still find your way, into the same valley, so that the number of choices that cells have is quite restricted. They can potentially be in these different states, but they're still in the same broad valley, and so they'll still be heading in the same direction and end up the same way. And that canalization is a way of, again, dealing with the randomness and the variability that we'll find at the very sort of lowest levels of the system, that you can have some noise and some variation, but you've still basically got the same kind of cell type.
Yeah.
And I think that that's an idea that kind of applies more broadly, and not coincidentally, in biology, in the sense that, I kind of think of it, for example, in terms of disease, that actually it's very striking when we think about it, that many different conditions manifest with similar symptoms. That was one of the problems during the pandemic, that we, found it very hard to know if we had COVID or if we just had cold or a flu. They manifested in, there was a lot of overlap with the symptoms. And that too, I think, is that there's a physiological canalization in the body, where different processes at the molecular and the genetic scale can have different physiological or phenotypic expressions. And there too, you get a kind of robustness, that you're going to get the same outcomes, the same sort of stable and, generally kind of desirable outcomes from different starting points.
Yeah. So the idea is, with something like 37 trillion cells or something in each of our bodies, that there's just so many possible forms that those cells can be in, but humans and organisms pretty reliably end up in these stable forms, because effectively what they're doing is rolling down an energy landscape. So like a big ball, or any ball will roll down a hill due to gravity, there's energy landscapes that mean it's more energetically what would you say, stable for the molecule just to be rolling down these energy landscapes and falling into these local minimas, which are physiological states like a cell lineage or any. But this I think this sort of extrapolates to all biological processes, right? And not just cell differentiation. That you can think of pretty much all biological processes as rolling down energy landscapes, finding stable energetic configurations for a little while until it's more energetically stable to roll down another hill and roll down another hill, and you're finding all these pockets. Is that sort of how you conceptualize biology and all of the different processes going on?
Yes, it is. I think we can see that this kind of canalization at many levels. So, for example, if we think about development again, there are certain stages that the embryo, certain sort of morphologies that the embryo has to go through. And what the most crucial of which, I guess, is often talked about is gastrulation, where there's a kind of a I suppose I kind of think of it as the moment when it stops looking like a ball of cells and starts to look like a little kind of embryo, that there's sort of a particular, tissue folding that goes on then. And again, cells have to get to that state. Sorry, embryos have to get to that state somehow, even if they're starting from, slightly different places and the cells won't all be in identical positions. The question often is, you talked about this being balls rolling down a hill, because they're sort of minimizing their potential energy. The question is often, well, what is the equivalent in biology? Are they minimizing it? What are they minimizing? Because we are systems that are inherently out of equilibrium, and we're dynamical systems. So we're not looking to get to our lowest energy state. If we were, that's being dead, basically. So, what is it what's the equivalent of gravity, if you like, or of, potential energy that is guiding these systems? What is driving this process towards these particular end states? And I don't think we have a very clear way of thinking, of conceptualizing that problem,
Interesting.
of thinking about, biology that way. If we think about it in terms of dynamical systems, then, we're thinking about it as an energy landscape, but it's not that. And it's what it comes down to is that there seem to be goals in biology. There seem to be things to aim for, and in the developmental process, the thing to aim for is the fully formed, properly formed organism. What is guiding that process at each step is still, a little bit hazy. We kind of think of each bit of the process in its own terms, but why it all sort of ends up, where is this ultimate goal, the shape of the organism, where is that encoded? If it's not in the genome, where is it? And I don't think we quite have the right story to tell about that at the moment.
Interesting. Maybe I had a misconception there. So what I had in my head is that a molecule or anything rolling down a hill in biology is moving from a less stable to a more stable state, basically electronically more stable state. There's that saying that science is just electrons looking for a place to rest. It's just more stable configurations of energy. Do you think that is incomplete? Is that not the whole picture? Is there something missing from that?
Yeah, there is something missing from that, because that is what tends to happen, as I say, in equilibrium systems. That or systems tend towards an equilibrium state, an
Yes.
energy state. Living systems don't. We maintain ourselves out of equilibrium. So we are these dynamical non-equilibrium systems where there's a constant through flux of energy and of matter, actually. And with that process, even for non-living non-equilibrium systems, things like hurricanes, for example, we don't fully understand that process of how systems out of equilibrium somehow organize themselves into these particular and these coherent structures. For living systems, as a whole, that's even more complicated, because it seems that there is this balance or this interplay or this dance between spontaneous self-organization and some kind of information that is encoded in the genome.
The Giant Shoulder mission is to explore radical ideas in biology, neuroscience, and consciousness, and elevate those stories to the highest possible level while keeping them accessible to everyone. If this interests you and you want to support independent science, then please consider subscribing to the Clips channel. Check out our 26 Neuroscience book. You can download it for free